was a button I needed to click. Thank you very much, and thank you for the great opportunity that you are giving me to talk a bit about the science that has been going on in my lab. Over the past 20 years, let's say, with a focus on the CRISPR genome engineering technology. So before I start, I surely would like to mention to which extent the scientific community is behind you, supports you in this terrible situation. I hope you and your family are safe and healthy, and the entire scientific community condemns heavily what you have to suffer from. There are no words, and actually it's a very awkward feeling to have to think that I'm giving this lecture in front of you who are in a very unpleasant and to say the least situation. It's really something that it's difficult to imagine. I'm located in Berlin, and we have seen for sure people from Ukraine coming right away to Germany. And it's very difficult to imagine that a war of such kind is going on next door. And I would like again to express all my support to you and your family and all the people of Ukraine. Actually, I'm going to start by, so for sure I cannot move, I cannot move my slides. I don't know if, I thought I could a little bit earlier, so I'm going to.
Maybe just escape and do it one more time, please. Yeah,
so maybe I stop sharing and I'm sharing again, and we're going to see how it works. That is the way often things go. Yeah, here I can move. Yeah. So actually, I'm going to start by my lecture by really emphasizing to which extent exchange of scientists is critical for research in our days, and that research is very international. And myself, I have actually I have had an international journey for the past now 25 years. And the project that I'm going to present you, the story I'm going to present you, CRISPR, actually originated in Vienna, and then continued in Umeå in Sweden, and then in Braunschweig in Germany, and now I'm based in Berlin. There had been key students and postdocs behind this discovery to support the research. And I would like to emphasize particularly Krzysztof Szyliński, a PhD student from Poland, and Elisa Delcheva, who was a master's student from Bulgaria. So just to show that the story I'm going to present has really an Eastern European spin, quite heavy. This is where my two students having really a key role in the development of the project. And now I've been working with really people all over the world, and I've always enjoyed this international feeling, and for the sake of science, and with really also the joy of the students and postdocs who have joined my team to really work together. So it's, I have always liked very much this enriching environment of internationality. And I do hope that in your career, whatever you choose to pursue, whether it's academic research or also research in industry or more applied research or a job around the science, and that you will really reflect on the necessity, I think, nowadays to have an experience, abroad, whether it's a short experience or a long experience, because this is always enriching. One sees a little bit how it's going on elsewhere, and it's also a chance to interact with people coming from different backgrounds. And I really think that this is really the key to enrich research and to lead to better development of research, and also surely more creativity as well. So having said this, beside the members of my team who were from different countries, there were also collaborations in Germany with a group located in Würzburg, in the US, with bioinformaticians located in Bethesda, and also collaboration with the group of Jennifer Doudna in Berkeley. And because I like to thank my research institutions is also important, because when you do research, you always have to also acknowledge the fact that there is a big machinery behind. I mean, I guess you are studying now and you realize also that there is a big machinery behind. And you understand even more while you are suffering from the war situation. So you have really a lot of people working on the infrastructure that allows you to have a roof to study and for us to do research. For sure, so beyond the fact that I'm presenting biological research, you have also a lot of people working on everything that is around infrastructure in terms of equipment. We work with very, very sophisticated equipment, and that requires also the expertise of mathematicians, physicists, chemists, etc., and other types of scientists who are really creating all these machines and equipment that allows us to really do even more deep and very sophisticated biological research. And you have all the personnel around the administration management that allows to support us researchers. So it's always big machinery, and I like to acknowledge this because it's important. It's also important for you because, again, I don't know to which extent you are you intend to pursue a career in science, but there are a lot of opportunities around the science to support research beyond the fact to be a pure researcher. And we are always happy to have this support. In Vienna, my research institutions were the University of Vienna and the Max Perutz laboratories at the Vienna Biocenter. In Umeå, this was Umeå University with the Laboratory for Molecular Infection Medicine Sweden. That is a Nordic partnership for molecular medicine with the European Molecular Biology Laboratory that is mainly based in Heidelberg, so EMBL. And I was part of a center that was called the EMEA Center for Microbial Research. And then in Germany, this was the Helmholtz Center for Infection Research located in Braunschweig with a medical school of Hannover. Now I'm at the Max Planck, and I founded about 3 years ago a unit that is called the Max Planck Unit for the Science of Pathogens, where the aim is to support research on pathogens that has been obviously also another threat, at least the last 2 years, with SARS-CoV-2 particularly. I've been working for a long time during all my career on bacteria causing diseases in humans, tackling different aspects of pathogenicity, whether it is to understand the mechanism by which bacterial pathogen become resistant to antibiotics or tolerant to antibiotics. Whether these are about studies with regard to deciphering mechanisms of interactions between the pathogens and their host, and are mainly focused on those pathogens that are really interacting with the human host, or whether it is also to try to understand how the host reacts to the pathogens and how they can put together this immunity against pathogens. And so for the past 20 years in my lab, we have mostly focused on the pathogen that is called Streptococcus pyogenes, also called group A Streptococcus. And some of you may have heard about this pathogen because it's very frequent in causing pharyngitis, for example, in kids. So with this pathogen group A strep, it also can cause skin diseases. And with regard to throat or skin diseases, it can be mild, but it can be also more severe. With, for example, necrotizing fasciitis, or also diseases that can cause sudden death, toxic shock syndrome. This is a pathogen that is very invasive, but 30% of us can be colonized with this pathogen without developing disease. And in my laboratory, we have always been interested in understanding the mechanisms by which Streptococcus pyogenes will cause diseases, will produce virulence factors. And the long term of this research was ultimately to find new targets for new therapeutic interventions, whether it will be the development of novel antibiotics targeting new types of molecules, or also understanding a little bit more about how our immune system can react against the pathogen, and whether there is also strategies of somehow therapeutics to be developed from this aspect. My approach over the years for my laboratory was to go quite deeply in the molecular mechanisms. Having had the experience during my postdoctoral time that this was the only way, doing really basic science, the only way to find novelties that then could lead to potential applications. The advantage also to work on bacteria is that they are a wonderful source, as well as viruses, of enzymes that have been largely developed in molecular biology and in genetics to perform genetics. Enzymes that can range from restriction enzymes that can cleave DNA and that have been used in the past very heavily for cloning, whether these are enzymes that allow to amplify the DNA, like all the polymerases that have been used for PCR reactions, or other types of enzymes such as ligases that allow to ligate pieces of DNA. So, or also enzymes that are used in reverse transcription. And particularly, the history has shown that this is really in the 60s, actually, that the research that was done trying to understand the interaction between bacteria and their viruses that are called bacteriophages. This type of research was very, very fruitful to find new enzymes, and therefore really start the field of molecular biology and genetic engineering. So working on bacteria, for sure, we are quite also very easily, we easily understand that the genetics is important because we have the possibility to manipulate genetically those bacteria. Sometimes it's difficult for Streptococcus pyogenes I had to develop some tools in my laboratory that will facilitate the genetics. So it's not as simple as it looks like, but nevertheless, because of their fast growth and the fact that we can grow them in vitro, a lot of technologies have been developed to genetically engineer not only Streptococcus pyogenes, but also other types of bacteria. The first one, E. coli, that is really the host bacteria species for molecular biology. But there are always a need for better tools, always. And this it's critical because it's now important to have better tools to be able to tackle research on higher organisms, which are, whether it's human cells or other model organisms, model animal organisms. So this, it's really critical. And when we work on with bacteria, we know as well that we have a chance to find those mechanisms. So that could lead to novelties in genetic tools. So this has always been the approach that I have been using in my lab. Also, we focused at some point on small regulatory RNAs, understanding that this class of RNAs had already a role as antisense in somehow the downregulation of gene expression, and that specifically Streptococcus pyogenes, which is not a very well studied pathogen, but my hope was to find interesting mechanisms for, again, the development of novel therapeutics to fight bacterial infections, and also genetic tools. With regard to actually the impact of the diseases caused by Streptococcus pyogenes, it's actually quite high. It's causing more than 500,000 deaths every year, and millions of people being affected with Streptococcus pyogenes infection. So it's really, for sure, it depends on which study you are looking at, but it's often ranked among the top 10 pathogens that are the most relevant to cause infectious diseases in humans. And it's a strict human pathogen. You will not find it in animals or elsewhere in the environment, which is a bit typical for example, Streptococcus pyogenes, or also Streptococcus pneumoniae, another Streptococcus pathogen, also called pneumococcus. So it's transmissible through among humans. So all those bacteria are very much encountering and subjected to stresses. And those stresses in the environment can be different, how do you say, different conditions in the environments, whether it's all of a sudden a high temperature or a cold temperature shock, or all the stresses that the pathogen can encounter in the human host, whether it's a change of acidic or environment. Any stress you can imagine is for sure very, very strong to take for a bacterial cell that has to adapt very fast to be able to survive. And that also coordinates the expression of the genes according to those stresses, so according to the environment, and also according to the growth of the bacterial cells. So you have a lot of fluctuations and a lot of regulations going on, ultimately leading to a differential production of virulence factors and all the factors that are important for the bacteria to be fit. And this involves a large number of molecules. So for sure, important, the molecules that are of protein nature, also of RNA nature. You have also small peptides, you have also metabolites that are very important. And in the laboratory lately, we have focused mainly on small RNAs. We also focused on other types of pathways, but CRISPR originates from the focus on small RNA molecules and all the proteins interacting with those small RNA molecules. When we started to work on small RNAs, actually, there was for sure a large body of literature, a large body of literature available, and that was showing that for sure, beside the RNA molecules involved in the link between the DNA that is transcribed into a messenger RNA that is translated into proteins, and also the ribosomal RNAs and the transfer RNAs important for the translation of this messenger RNA into proteins. There was another class of molecules that that had already started to be studied in the 80s. And these are RNA molecules with regulatory functions. So they are called small RNAs, regulatory RNAs, non-coding RNAs. So they are different types of names, but in principle, those RNAs do not code for proteins. I mean, some may code to very small peptides, but most of them are in this classification are non-protein coding RNAs. And they can interact with the expression of the messenger RNA through different ways, often by base pairing, so by somehow finding a kind of a base pair complementarity with messenger RNA target. And by this can either affect the stability of this messenger RNA or can affect the translation of this messenger RNA, activate or inhibit the translation of this messenger RNA. And they can also interact directly with proteins. And when they interact with protein, they can block the function of the protein. So they sequester proteins from their natural functions, or they can activate the functions of the proteins. What was not really known at the time, so about now 15 years ago, was whether there are some RNAs that can interact directly with DNA and affect the fate of the DNA. And this is what CRISPR brought. So the CRISPR technology originates really from understanding at the basic level the CRISPR-Cas mechanisms existing in bacteria and in archaea. So for sure, my team is not the only one to have studied CRISPR. Actually, when we started to work on CRISPR around 2006, 2007, a large number of scientists were already working on these systems, which they had discovered in bacteria and archaea. I don't have enough time to go through the entire history, but it started in the 80s with the discoveries of some specific signatures of this CRISPR-Cas system that are called spacers, and that have as origins mobile genetic elements type phages, the viruses of bacteria, or also plasmids that are mobile genetic elements existing in bacteria and also in archaea. So the CRISPR-Cas9 technology is really composed of an RNA component and a protein component. So what you see in black is a target DNA. And the idea is really to have harnessed this CRISPR-Cas system existing in bacteria into a genetic engineering technology. So technology that in principle can recognize a certain sequence of interest on the DNA and will be able to thanks to this CRISPR-Cas mechanism, to cleave at this specific sequence on the DNA. And when the DNA is cleaved, then, and specifically in higher organisms, then there would be, how do you say, there will be the repair machineries of the higher of the cell of the higher organisms being triggered, allowing to repair these DNA fragments that have been cleaved. And depending on how you design the technology, the repair of these cleaved fragments at the site of interest can then lead to really genetic engineering, in the sense that at the site of interest, you can either introduce a mutation on the DNA, you can correct a mutation on the DNA, you can introduce a piece of foreign DNA, you can replace a gene by another gene. So it's very powerful. And this consists of, for the CRISPR-Cas9 system, of one protein that is called Cas9 that has enzymatic properties, and that is associated, so really bound to an RNA that is in nature a duplex of two RNAs. One is called CRISPR RNA, the other one is tracer RNA. And for the purpose of the technology, those two RNAs that are linked together naturally were further linked with the addition of a linker loop to really create one single RNA. And the idea is that the single RNA has a complementary base pair complementarity with the target DNA. So one just needs to design this RNA molecule in a way to really have the sequence of this RNA molecule that can base pair to the sequence of the DNA of interest. And then this RNA by base pair complementarity can bring this enzyme that is bound to this RNA to the site of interest, and this enzyme will cleave. And as I said, the host cell will repair, and one can then trigger different aspects of the genetic engineering. So I will come back very briefly on the evolution, but I just want to say that when we published actually an article in Science that was the second article describing the end of the CRISPR-Cas9 mechanism to target the DNA. This was already now 10 years ago, and very fast the scientific community has really picked up on this technology and has really derived different versions of the technology. So from the version 1.0, multiple versions have evolved since 1.1, 1.2, 2.0. So, and very interestingly, actually combining this technology with other genetic engineering technologies which were discovered prior to CRISPR-Cas9, or which have been discovered since, to really enlarge this genetic toolbox and giving actually a lot of power to these CRISPR technologies. With regard to the history, very briefly, it's important to really put into context the technologies. So again, I talked to you at the beginning of my talk of the 60s. But here I would like to really mention that for sure in the 19th century, and this was really the first start of understanding of genetics through the origin of species, the laws of inheritance. Then the DNA that was isolated, the fact that it was shown to carry the genetic information, the structure of the DNA double helix that was determined, the genetic code deciphered, and then really the 60s and 70s started really this era of development of genetic tools. CRISPR-Cas9 came on board 10 years ago. And this was following the engineering of artificial enzymes based on the property of naturally existing enzymes that are called zinc finger and talon nucleases. And these engineered zinc finger talon nucleases actually were also used prior to CRISPR-Cas to really perform clean and very targeted gene editing. So the results in a way is the same, they work differently. The programmability of the zinc finger and talon nucleases are on the side of the proteins. One has always to engineer a new protein for a new site on the DNA of interest to be manipulated, whether CRISPR, whereas as CRISPR-Cas9, it's more simple because the programmability is on the side of the RNA. One changes the RNA and then can therefore apply the technology to diverse sequences of DNA. Very fast, I will go through the history. So from the side of my laboratory, the interest to work on CRISPR, as I said, was because we were interested in finding new small RNAs in Streptococcus pyogenes. And in 2006, we performed a bioinformatic analysis. And we found actually these CRISPR RNAs, these RNAs that were actually, and together with also CRISPR-associated proteins that were already annotated as a CRISPR system. So this was already mentioned by colleagues of mine. Interestingly, in the vicinity of this CRISPR system, so as a matter of fact, the CRISPR-Cas9, that was given the name after Cas9, but went through different nomenclatures, there was a piece of DNA that seemed to encode small RNAs that was typically small RNAs, not coding for proteins, and that ultimately we call tracer RNA. And here you can see northern blot analysis of its expression throughout the growth phase of Streptococcus pyogenes, so expressed in different forms in a quite constitutive manner. With regard to CRISPR-Cas, we figured out reading the literature that this was a system, a defense system in bacteria that was allowing bacteria to defend themselves against mobile genetic elements, type plasmids, transposons, and phages that are the viruses of the bacteria. So for sure, bacteria, when they are encountering viruses, they have to deal with either lytic phages that are going to kill the cells, or lysogenic phages that can bring new genetic traits to the bacteria. For sure, if the bacteria encounter a lytic phage that is really is meant to kill the bacteria, they really have to defend themselves against this. If it's about a phage bringing new virulence factors, maybe they don't want to acquire these virulence factors. This is the case of Streptococcus pyogenes, being very much involved with viruses that upon infection of Streptococcus pyogenes can actually bring a new virulence factor to the bacteria by just integrating the genome of the virus into the genome of the bacteria and make it part of the genome of the bacteria. And so there are different defense systems, immune systems, which bacteria and archaea as well have evolved over time. Restriction modification systems is one of those defense systems, and led actually to the discovery and development of these restriction enzymes used for cloning. CRISPR-Cas has a particularity to be classified not as an innate immune defense system. Sorry, I have to stop for 1 second and I'm coming back. Briefly. Okay. Sorry, because I was disturbed, but now I'm back on track. CRISPR is one of such defense systems, and it has a particularity to be an adaptive immune system. So it's quite complex in the sense that when bacteria encounter phages, in this case, there is a step of recognition whereby the bacteria, so the CRISPR system, can recognize the invading phage DNA and will cleave some pieces of the phage DNA to integrate it into the CRISPR array. And here you see the different sequences of colors in the CRISPR array. This corresponds to different types of phage or mobile genetic DNA that has been incorporated upon infections. And in the vicinity of this CRISPR array, you have the CRISPR-associated genes that code for the CRISPR-associated proteins. And this array of spacers are also interspaced by very short sequences that are called repeats, and that are integrated in between those spacers sequences of mobile genetic element nature. Then the CRISPR array will be transcribed into a long precursor CRISPR RNA molecule that will undergo processing to produce this mature CRISPR RNA that are very small in sizes, and that will have the particularity to associate themselves with a complex of CRISPR-associated proteins. And thus this machinery of RNA and CRISPR-associated proteins will be able to recognize the phage DNA upon a second infection and destroy the phage DNA. And then this is the end for the bacteria, for the virus. The viral DNA is cleaved and it cannot propagate, cannot kill the bacteria, or cannot even integrate into the genome of the bacteria. So this is a dead end for the viral DNA. So, and the virus itself. So this was the dogma when we started. Interestingly speaking, this system has largely evolved. And what was known is that the RNA work with, the CRISPR RNAs really work with a complex of CRISPR-associated protein. For CRISPR-Cas9, we showed something differently. What we showed is that there was only one CRISPR protein involved in the step of recognition of the phage DNA upon a second infection, and that instead of one RNA, one CRISPR RNA mature, there were a duplex of RNA, CRISPR RNA, tracer RNA. So this was really through for sure, a genetic and biochemical approach that started in my lab in 2007 or so. And that led to 2 publications in Nature and Science, and with finally the understanding of the mechanism and an understanding of how we could simplify the mechanism for an RNA programmable gene editing tool. As I said, it can correct, delete, add, replace genes. Very, very fast, it has been shown that it was working extremely well in different types of organisms, model organisms, like, for example, mice, fish, Drosophila, also plants, organoids, and human cells. And so this was really in need because it allowed to simplify heavily the genetics over the past 10 years, sorry. The CRISPR-Cas9 system itself has largely evolved. So nonetheless, it's part of the multiple systems existing. And here is just a slide showing you to which extent the CRISPR-Cas systems have evolved. The CRISPR-Cas9 is type 2 system, so located on the left with different subtypes that have been described since the how do you say, the description of the CRISPR-Cas9 system. Interestingly, the Cas9 system itself has evolved largely among bacteria, so it's mostly present in bacteria, and it has largely evolved with the protein Cas9 having also large diversity in sequence, as well as the CRISPR RNAs and the tracer RNA that have a large diversity of sequence. And it's really through looking at the evolution of those systems, and you have for sure understood it with SARS-CoV-2 that evolves in multiple variants. CRISPR-Cas, it's also the same. This is the pressure of always the arms race between bacteria and phages that allows those different systems to largely evolve. And the interesting aspect of this project is that key for my lab to really understand the mechanism was really to look through the diversity of those systems and to try to find what would be conserved. And what we figured out is that specifically this key element, the tracer RNA, was actually an RNA molecule of a specific family that will be defined by the ability to interact with the CRISPR RNA, but no specificity no other specificity of sequence nature or structure. Really, the key point is a tracer RNA that can be different in sequence depending on the multiple bacterial species, but that will have the ability to interact with the repeats of the CRISPR RNA, and this dual RNA being able to bind the protein Cas9 and then being able to target the phage DNA to cleave it. I think I have to look at my slides. Maybe this was the end of my slides. I think I have to check. Yes. So I was back with, no, sorry. I was back with another slide that is, that I had difficulties. Okay, so with regard to the evolution, something that I wanted to say as well is that in addition to the CRISPR-Cas9, other types of CRISPR-Cas systems have been evolved, have been developed as also other CRISPR-Cas tools, specifically from the type 5 and type 6 that are relatively minimal and that involves Cas proteins that can interact with the RNA. So not only So systems can act on the DNA of the phage, but also can act on the RNA of the phage. And this also is also harnessed to try to develop those technologies against DNA and RNA of interest in any types of cells and organisms. The applications have been really huge, and a lot of scientists, clinicians, and developers and people working in biotechs and industries have really picked up on this tool and developed it further as a tool that really makes the difference in the production of clean plant crops. That also makes the difference really in everything that is research and development by facilitating and speeding up the engineering of genetically modified organisms. Also the possibility to develop screens, which is very important for finding new targets for therapeutics or for developing models that are important to test therapeutics in development. Also for transplantation in pigs. So you have a lot of development that is still going on, and is expected to still go on for the decades to come, with also the challenge, but which has shown some really success lately in clinics to really combine this genetic engineering technology with cell therapy. And that is, for example, for certain blood disorders such as sickle cell anemia or beta thalassemia, the possibility to really engineer, thanks to CRISPR, blood cells that can then be used to repopulate the blood cells of the patient, and that can provide a cure for certain types of diseases that have a genetic background in blood cells. I will finish my talk by showing you an electron macroscopy picture of Streptococcus pyogenes, and just tell you that actually in my lab we are mainly working on other types of mechanisms than CRISPR now, because I'm interested in knowing further about Streptococcus pyogenes, and again in my pursuit and interest in finding new types of mechanisms other than CRISPR-Cas that could have also some useful application interest. And I think it is specifically important to continue to really study the field of macrobiology. I just want to say this because you have seen it with SARS-CoV-2, with the pandemic that we have experienced, and you have for sure a large number of other pathogens that are evolving quite fast, and so really actually a urge to really continue to work on these pathogens, and to also train the young community of scientists to be interested in those pathogens, because this is maybe what your generation will have to deal with even more. And so there is always a need to really have this knowledge to be able to anticipate and develop better tools to fight these pathogens.
Excuse me, Professor, we cannot see a picture you wanted to show us.
So maybe because I did not share the screen?
Yeah.
Or, okay. Yeah, so this was, I guess you see it now. Is it working?
Yep.
So this is, the picture of Streptococcus pyogenes electron macroscopy picture. So I would like to end my talks as such, because I did the acknowledgement at the beginning, and I would like to thank you for your attention.
Thank you very much for the great lecture. It was a great pleasure to talk with you today. I hope that very soon that we will meet in peaceful Kiev, and I hope that your lecture will be in person in maybe in one of the biggest universities.
I hope so, and I really hope so. And again, I really hope that this is going to end soon.
Thank you very much.